Okay, my name is Jeff Horsburgh. I'm at the Water Lab, Civil Environmental Engineering, like I said. And before I get started, there are other names on these slides in addition to mine. Sorry, David, your name kind of got split between two lines, and that's because Amber's name got longer since the last time I presented this slide. So I want to acknowledge Amber Spackman-Jones, who was a graduate student, got her master's degree here. She did a lot of the work that I'm going to talk about today. And of course, David Stevens, David Tarbotton, and Nancy Mesner was a part of a lot of this work as well. So I'm going to talk today about some of the work that we've been doing in the Little Bear River. In our proposal that we wrote, we talked a bit about the Little Bear River test bed, the environmental observatory work that we were doing there. So I thought I would introduce you to kind of the landscape, the types of data that we've been collecting, some of the things that we've learned, and we'll go from there. Okay, so the objective of my proposal today, we want to look at some of the monitoring work, and then also at the end I'd like to kind of draw it to some conclusions and some discussion points about how the types of data that we're collecting with respect to water quality either enable or don't enable us to examine human hydrologic, engineering, and ecological domains. Okay, so my favorite place in the world, not really, but it's nice to work near my home. The Little Bear River is here in Cache Valley. It's the south end of Cache Valley. It drains a good chunk of the south end of the valley. When we started working in the Little Bear, our objectives were a couple of different things. One, we wanted to look at what the observing infrastructure, what would be needed if we wanted to make high-frequency estimates of things like total suspended solids and total phosphorus, which are water quality constituents that we are interested in, but with high frequency. We typically don't measure these things with high frequency, and by high frequency I mean on something less than a day time scale, as opposed to weeks and months between measurements. And then our other objective was, once we start collecting a lot of high frequency data, what kind of infrastructure do we need to manage those data? What kind of sensor infrastructure do we need to actually collect the data? What kind of database and cyber infrastructure do we actually need to manage the data that we're collecting? Okay, so if we take a look at some of the water quality issues that we're interested in here in Cache Valley, and I think this is probably true of a lot of places in the western U.S., we have, nutrients are an issue, nitrogen and phosphorus, and that's primarily because of eutrophication in downstream water bodies. You put nitrogen and phosphorus into Cutler Reservoir. It does the same thing in Cutler Reservoir as it does to your lawn when you put fertilizer on it. It grows lots of nice green stuff, and it actually isn't just a problem in the reservoir. This is the Little Bear River near Wellsville, and this is Amber pulling this nice mat of awful, disgusting, smelly algae away from our water quality sensors because they were kind of growing up around the pipes that had our sensors in them. So this is not an isolated thing. This is something that actually happened. So pollution sources, if we look around us, I think agriculture, not to pick on agriculture, but we live in a predominantly agricultural area, and we have animals that do what animals do. We have a lot of streams that look kind of like this picture in the middle where we have stream banks that are not in the best condition. And then we have agricultural fertilizer, pesticides, all sorts of things like that. We also have, not to a great extent in the Little Bear River, but I didn't want to discount this because just over in the Spring Creek, which is the next drainage to the east, and then of course the Logan River, Blacksmith Fork, we have a lot of urban stormwater runoff with the cities around in Cache Valley. This picture is interesting. This is not a beautiful, well-manicured lawn. This is actually duckweed growing in the sewage treatment lagoons out in Wellsville. And these nice trees kind of going around the outside of there, that's actually the river going right around the outside of the sewage lagoon. So we do have wastewater treatment facilities here in Logan, and they do discharge to our water bodies. And then, of course, industry, not so much in the Little Bear, but again, we do have industrial sources here in Cache Valley. So when we go and we want to understand water quality, the things that we're interested in are things like, what are the sources of pollution? How much is coming from each source? And those are things like transport pathways. How do the pollutants get into the stream? How do the pollutants get into the water bodies? And then the fate, we're also interested in once those pollutants get into the stream or into a water body, what happens to them? How long do they stay there? Do they settle out? Are they taken up by plants? We're also very interested in how water quality changes in space and time, and I'm going to talk quite a bit about this. But in response to natural events, so things like what are the seasonal differences, what happens when it rains, what happens when the snow melts in the spring, and then in response to human events. So what happens when they open the gates on Hiram Reservoir and start blowing water out in the spring? What happens when they turn on the agricultural diversions or shut them off? And what happens when we have large return flows or lack of return flows in different parts of the system? Okay, so the space challenge. How do water quality conditions vary throughout the watershed? We'd like to look at what are the natural hydrologic features. So tributaries coming into the main stem of a river. You know, the land use in the Little Bear River changes as you go from the top to the bottom. We go from predominantly forest and rangelands to agricultural lands lower in the watershed. Management practices. Doug probably will tell us about this eventually, but there has been a lot of work done in the Little Bear River watershed in implementing agricultural conservation practices on farms with producers in the watershed to actually improve water quality. So what are the results of those management practices? And of course, what processes, human and natural, drive the variability? Is it more natural processes, or is it the human things that are going on that's really driving the variability that we see in water quality? Okay. So the Little Bear River watershed is on the order of 740 square kilometers. When we talk about water quality monitoring, the first thing that we do when we go to do a watershed study is we go out and look for existing data. We come to this watershed, and if I were to go to EPA's STORET database and say, give me all the water quality data for this watershed, I'd look at the river and I'd see on the order of 45 monitoring sites. And those are the red dots on the river there in the Little Bear River. I might say, wow, I've got 45 sites. If I want to characterize the spatial and temporal variability in water quality in this watershed, there's really only 2 sites that really fall into what we might call a data-rich category. And I'll tell you, I think let's qualify data-rich. So diversity of measured variables, longer period of record, you know, these 2 sites are kind of on the order of 30 years or so of data, and a large number of samples. And to qualify that, looking at the table over here, this is 10 years worth of water quality samples for, I think this is total phosphorus in the Little Bear River, which is a pollutant of concern in the Little Bear River. We have, you know, less than 10 observations of stream flow, and on the order of, you know, 10 observations per year of total phosphorus. And this is kind of the typical water quality monitoring data that we have to assess through the whole watershed spatial and temporal differences in water quality. So there's some obvious issues there. And it gets worse when you talk about the time challenge, because we want to know how and why does water quality change over time scales of hours to years. We know a lot of water quality processes are dynamic and have time scales that are far shorter than monthly or even annual. And then we'd like to know whether water quality conditions are getting better or worse. And even more, we'd like to know what might happen in the future given changes that might happen in Cache Valley. I put the climate change here, but I think it's more likely that we're going to have significant land use change, and then climate change is this big question mark hanging out there. What's going to happen? You know, how is that going to affect things? So to address this, we started. We proposed to the National Science Foundation to build a bit of an environmental observatory in the Little Bear River watershed. And what that resulted in was a network of stream monitoring sites. There are 7 of these, and you can see the variables that we collect continuously on time scale of, I think they're half-hourly data. And then there are 4 weather stations, which have a suite of meteorological variables, and then soil moisture and temperature at 5 different depths. They're all connected via spread spectrum radio telemetry network. They all send their data back to the Water Research Lab every single day, so we're getting this data daily. And our objective was, let's collect data at a much higher frequency, and then let's see if we can start addressing some of these deficiencies in the data sets that we've got with higher frequency data. So if you look at 7 water quality and stream flow monitoring sites, 4 weather stations, collecting data for this number of variables on a time scale of half-hourly, you don't go very long before you run out of room in Microsoft Excel to put your data. So obviously we needed some infrastructure to manage the data. And I have 2 token cyber infrastructure slides. Cyber infrastructure was in the title of my presentation, but I have 2 token slides. We built a system that enables us to essentially in real time have connections to all of our monitoring sites, send those data back to a base station computer that's at the Water Research Lab, load them into a database that's on a server there, and then they're essentially immediately available on the internet once they've actually been loaded into the database. We have trouble with our batteries in the winter, so the data only get downloaded once a day, but that's good enough for what we've been doing. We've also worked on a more national scale with the Consortium of Universities for the Advancement of Hydrologic Science Incorporated. COASI is the acronym for that. But there's a large-scale cyber infrastructure project where we've been working on cyber infrastructure systems to share the data that we're collecting on a national scale so that other people can discover our data and find out what we're doing and the data that we're collecting. And my point is not to go through all of these boxes and arrows and all that kind of stuff, but to get you guys to understand, we are making efforts here at Utah State University to do this kind of work, and it's ongoing. The Little Bear River is one of our test beds for that kind of work. Okay, so getting back to Amber Spackman-Jones. She spent most of the time on her master's thesis while she was here working on the continuous water quality data that we are collecting. And she spent a lot of time developing approaches for correlating our continuous turbidity measurements, measures of the water clarity, with things like concentrations of total suspended solids and concentrations of total phosphorus. And those relationships over there kind of show you at one of our sites, the nature of those relationships. They're typically pretty linear, and they're actually fairly good at most of the sites in the Little Bear. But the reason why she spent so much time on this is because we have continuous data now for turbidity. And if we have these relationships between turbidity and things like total phosphorus and total suspended solids that we're actually interested in, we can take that relationship and transform our continuous turbidity into continuous total suspended solids, continuous total phosphorus. Now, the point of this slide is if we take our continuously estimated total suspended solids and we start kind of artificially decimating those data, once we get down to the weekly and monthly time scales of most water quality data that are out there, you can see that we almost completely lose the signal of water quality in our watershed. So sampling frequency is really important. And on the right-hand side over there, what you're seeing is the result of, we took the continuous data and calculated an annual load of total suspended solids and total phosphorus. And then we started again artificially decimating the data, and we made estimates of the total suspended solids load and total phosphorus load based on hourly data, daily data, weekly data, and monthly data. And what you can see is that you don't lose much resolution if you go from half-hourly to hourly, but when you start going to daily, weekly, and monthly, you start underestimating the loads that you're getting, and that's because you're taking away all of the peaks in the data, and you're left with, this kind of stuff. You might get really lucky and get one of those peaks, but you're not very likely to get those peaks anymore. Another reason why this is so important is because if we look really closely at the discharge and say our continuous estimates of total suspended solids in this watershed, and this bottom plot right here shows essentially the cumulative percentage for 1 year of the total suspended solids and total phosphorus loading at the Paradise site. About 50 to 60% of that annual load is moving through the watershed in about 3 to 4 weeks of the year. So typical water quality monitoring, you might get 1, maybe 2 samples in there to characterize that whole time period where 50 to 60% of your constituent loading is actually moving. So the high frequency data actually allow us to characterize that. Now this also gives us information about potential flow pathways that the pollutants are getting into the stream. So for example, I know that particular time of the year is when the snow is melting. It's early spring snow melt. And this doesn't give me all the information that I need, but I know that if I want to reduce the amount of total suspended solids and total phosphorus getting into the stream, I would do a lot better to target the mechanisms that are happening during that particular time than to look at, say, summer storms or something like that, which show up as little teeny blips in our time series. Okay, so the time challenge also includes variability across water years. This compares discharge, total suspended solids estimates, and total phosphorus estimates for 2006 and 2007 at one of our sites. And you can see 2007 was almost non-existent in terms of stream flow. We didn't get as high of peaks in the TSS and total phosphorus, but if you looked at the loadings in 2007 would be a lot lower than 2006. And this is pretty common in our western watersheds where we're totally driven by the amount of snowmelt that we receive. Okay, so how do human activities then affect water quality conditions? Obviously, what we're measuring in the stream is kind of an amalgamation of everything that's going on because the stream is an integrator of the entire watershed. But here's an example of water quality data from the Little Bear River at Wellsville. It's the same site where you saw Amber pulling the big mat of green nastiness out of the stream. But this is in December and January of which year? Did I put the year up there? Sorry, I would have got a bad grade in class because I didn't put the year up there. But I think it's probably 2007. But what happens is at that time of the year, the reservoir operation rules for Hiram Reservoir change, and they start moving water out of Hiram Reservoir. So up here is the discharge in cubic meters per second. On this day, they all of a sudden open the gates at Hiram Reservoir. And so we go from this nice diurnal pattern of dissolved oxygen to basically nothing, dissolved oxygen variations. The pH variations go away, not completely go away, but it totally changes that. And our specific conductance goes way down because now we've completely changed the source of our water from local groundwater sources with the gates of Hiram Reservoir shut to reservoir water screaming past. And when I saw these data, I thought, oh, something's gone wrong with my sensors. So we went out and we looked at the stream, and sure enough, the water was deeper. And instead of seeing that nice green, nasty stream, we saw a nice, clean channel that was about twice as deep, twice as wide, and all the gravel were nice and clean. And it took a while for all of those organisms that are generating these diurnal variability in water quality to actually recolonize and come back. So there's an example of how one change in management completely changes the water quality regime. Okay, this is an example of taking those same kind of total suspended solids estimates over a year, and unfortunately, I didn't switch these, but we have the red dots represent the annual loading of total suspended solids that we calculated for these sites in the watershed. And the size of the dot is proportional to how much loading there was. And so you see, we have this little dot right here, which is the South Fork. We have a bigger dot right here, which is the lower South Fork, which is where Davenport Creek comes in. We get bigger at Paradise, and then we have Hiram Reservoir. And all of a sudden, that dot gets much smaller. Bad news for Hiram Reservoir, because all that red dot has to go somewhere, and so it's settling out in Hiram Reservoir. But interestingly enough, the dot at Mendon, which is at the downstream end, just upstream of Cutler Reservoir, we've significantly grown that dot again. And if you look at the cumulative plots of discharge and total suspended solids, in the upper watershed sites, the total suspended solids doesn't always follow the flow. So these dotted lines are the flow, the solid lines are the total suspended solids, and these gaps mean that the total suspended solids is moving through the watershed faster than the flow. But by the time we get down lower in the watershed, this is Mendon, they're almost the same. So the structures that we have in our watershed, the management of our watershed, changes the way constituents are moving through the watershed. Okay, David's done a lot more work on this than me, but we've been looking at conceptual models of things like how does the dissolved oxygen diurnal variability happen and what causes it, and what are conceptual models of why it occurs. And so this is one conceptual model that shows a bunch of variables that say, okay, I get my minimum dissolved oxygen in the morning, I get my maximum in the afternoon when the plants are growing because we have photosynthesis and respiration, reaeration, and all these processes going on. But if you look really closely at the dissolved oxygen data, those curves don't all line up with the same timing. They're not all the same magnitude, and they don't all have the same kind of baseline. And so the questions are things like, Why are there differences in behavior based on where we're at in the watershed? What causes the difference in the shape of the diurnal DO curve, the timing of the diurnal DO curve, and those offsets from each other? Okay, so to get to some of the discussion then, the data cube. We tend to represent our water quality data in a data cube, and that means any particular data value is a function of where it was measured, what it actually represents. So this is a measurement of stream flow or dissolved oxygen, and then when it was actually collected. And that works really well for us. It works really well in databases to represent data that way. But the problem with that is all of the additional information, the going out in the watershed, the seeing the stream, the understanding the mechanisms is in my brain and it's not encapsulated with the data. So all of the additional human dimensions that we want to represent, things like what was the reservoir release status when I made this measurement? What were the diversion status? Were they irrigating? Were they not irrigating? What's the local hydrology? Were the soils saturated? Were they not saturated? What conservation practices have been put in place upstream? We don't have good representations of those kinds of data. Other opportunities are things like we don't always capture the results of all types of studies. And there are other people doing work in the Little Bear River watershed that I don't even know about. And so, I might want to also know things like what is the algal biomass in the stream, and that affects water, dissolved oxygen and those kinds of things. And there might actually be somebody else out there measuring that would allow me to incorporate that into the model that I'm looking at. So I think there's some real opportunities in getting all of these groups, us together in one group, to start putting more of the pieces together. Okay, so sorry about the text being so small on this slide, but discussion and conclusions, it's really difficult to understand and manage what we don't measure. And it's pretty obvious when we start looking at the types of data that we have available to us. If we're not making any new measurements, it's pretty inadequate to do the things that we want to do if we're looking at changes over space and changes over time. We need data that are consistent with the time scales of the processes that we're studying. If we're looking at diurnal variability and dissolved oxygen in a stream, we better be collecting data with time scales of minutes to hours and not days. We think about what should we be monitoring. It's great that we're going out and collecting data on flow volumes, pollutant concentrations, mass loadings, but I think we need to broaden the scope and go beyond just space and time. We as engineers tend to kind of focus in on things, but there are entire fields of study that we don't do well with, we don't address, and we certainly don't know as much about. Ecological data, geomorphological data, management behavior, and I put in the management behavior category things like reservoir operations, agricultural, water system management, water quality regulation, and then social science data. This is sort of an arbitrary division, but the individual behaviors. What are the behaviors of the producers, the behaviors of the water delivery organizations? And I think we definitely, in putting all this together, and this goes back to some of the cyber infrastructure work that we're doing, we need to design structures for representing and synthesizing diverse data. It's a real challenge to put all of these types of data together. And something that David Stevens and I discussed as we were talking about what we were going to present today, this last one might be a good discussion question, but it's how do we design a monitoring program for what we will need in the next 30 years as opposed to what we didn't have over the past 30 years. So it's easy for us to look back and say, well, we wish we would have this, and we wish we would have had that, and we wish we would have had that. But I think more proactively, if we're designing scientific studies that are going to help us really understand what's going on, we should be looking forward and say, we need this to answer this question, we need this to answer that question, and then we go out and get that information. So I've probably taken twice the time that you wanted me to take.